Good morning, dear students of class 12 science. Welcome to biology class. We are still dealing with the chapter strategies for enhancement in food production. So today we are going to learn about plant breeding for disease resistance. Then we will discuss about mutation breeding and plant breeding for developing resistance to insect pest. Then in the next class we will cover the topic biofortification, plant tissue culture, and single cell protein. Okay, so first topic of the day is plant breeding for disease resistance. Clear? So mainly the different types of crop plants in different climatic conditions are attacked by the fungus, okay, by bacteria and by the virus. So these disease causing organisms, they're usually called the pathogens because they used to cause different types of diseases in case of plants. Clear? So these particular pathogens, or let's say these causative organisms, and the different mode of transmission of the diseases are: number one, let us see, okay, the different type of diseases which are caused by fungi. First one is brown rust of wheat, red rot of sugarcane, and the late blight of potato. These are the diseases which are caused by the fungi. Diseases caused by bacteria, we can see black rot of crucifers. By the viruses, we have got tobacco mosaic virus and the turnip mosaic virus. Clear? So breeding and development of disease resistant cultivars or variety of crops enhances food productions and reduces the dependence on the fungicides that is mainly used to kill the fungi, and the bactericides that is mainly used to kill the bacteria. Okay, fungicides are mainly used to kill the fungi and bactericides are mainly used to kill the bacteria. So incorporation of disease resistance is the only means of developing preventive measures or developing resistance against the diseases like viral diseases, okay, or the diseases caused by bacteria or the diseases caused by fungi. So we have to incorporate the disease resistance property in the crop so that they can be free from the attack of these particular causative organisms or the pathogens. So therefore, disease resistance is the ability of the plant to remain unaffected even when attacked by the pathogens. So whenever the plant which has been incorporated the disease resistance property, whenever they are attacked by the pathogens, they will remain unaffected. So that property is called the disease resistant property. And when the host genotype do not allow the pathogens to grow and flourish over them, they form resistant host line. So those disease resistance plant, they will form the host lines. Okay, so therefore remember kindly the type of diseases which are caused by the fungi, the diseases caused by the bacteria, and the diseases caused by the viruses. Now how can we incorporate disease resistance property in a particular crop or a plant? Okay, so we have got different technique or the different methods by which we can incorporate disease resistance property in a plant. So first one we'll discuss about is conventional breeding technique. Now in this technique, what we do, alright, so this particular method of disease resistance incorporation in plants is mainly composed of hybridization and selection. So there are mainly two techniques, alright, under this one, under conventional breeding technique, two steps we can say. First is hybridization and number two is selection. This particular topic we have done already in our previous classes. So the steps which are mainly under these conventional breeding techniques are: number one, screening germplasm. You know what is germplasm now, okay? Then number two, hybridization of selected parents. We have done about hybridization already. Number three is selection and evaluation of the hybrids, followed by testing and release of new varieties. So these are the steps under the conventional breeding techniques by which we can develop the disease resistance crop. Okay, so let's see the different types of disease resistant crops which are developed by this conventional breeding technique. So this is very important. Clear? You have to remember the name of the crop, the variety, and resistance to diseases. So first one is the crop wheat. It's in the page number 515. Clear? So go through it. Page number 515, table number 9.1. One more time, page number 515, table number 9.1. All right. So first crop is wheat. The variety is Himgiri. Resistant to disease is leaf and stripe rust and heel burnt. Number 2, brassica. The variety is Pusa soranim. Resistant disease is white rust. Then we have got cauliflower. Variety Pusa supura and Pusa snowball K1. Resistance black rot and curl blight and black rot. Then cowpea. Variety Pusa komal. Resistant disease bacterial blight. Then chili Pusa sadabahar. Resistant disease chili mosaic virus, tobacco mosaic virus, and leaf curl disease. So kindly remember the variety of the crop and the resistance to diseases. Okay, so this is mainly constrained in conventional breeding technique. All right, okay, now next one we'll go through the mutation breeding. Now before going into the mutation breeding, what do you mean by mutation? You're doing class 10 also. Just briefly, all right, try to recall what do you mean by mutation. Clear? We are not going, we're just going out of the topic now. Mutation means just think we have got the DNA sequences. We have got adenine, thymine, guanine, and cytosine, ATGC, right? Now, in our DNA sequences, if any one of the base sequence will be changed, right? If any one of the base sequence will be changed, then that is called mutation. Okay, so mutation means changes. Clear? Changes that occurs within the base sequence of our DNA. Okay, so same changes can be done in case of plants also, in order to promote breeding in plants. Okay, so in order to develop the disease resistant plants. So now we'll just talk about mutation breeding. So mutation can be introduced or can introduce certain genetic variation. Genetic means gene. Okay, in the hereditary material. Hereditary means the characters which are inherited from parents to offspring. So those genetic changes can be induced in the plant, and those changes can be inherited from the parents to the offspring. A change in the base sequence within a gene can produce a new character in the parental type. So if there will be a change in the base sequence, means remember carefully, adenine, thymine, guanine, cytosine, these are the base sequences, right, in our body, in the DNA. So if a base sequence will be changed within a gene, then that will produce a new character, a new phenotype, means the character which is visible from the outside. So new characters will be produced within the parental type due to the mutation breeding. Okay, so therefore, since mutations are able to produce new changes, they are able to bring new characters. So this type of changes can be brought in plants for the benefit, right, in order to develop the plant which are resistant to diseases, resistant to pest. So we can bring or we can introduce these mutations in the plants also. Clear? So look at the second paragraph. So mutations can be introduced experimentally through the use of chemicals or radiations like gamma ray. Okay, and such mutations which are induced, which are introduced experimentally, they are called induced mutation, because we are inducing the plant to undergo mutation. So such mutations which are introduced experimentally in the plant are called the induced mutation, and the use of this induced mutation in the plant is called mutation breeding. Okay, so therefore the technique of using mutation in the field of plant breeding in order to develop disease resistance plants, or in order to develop or incorporate genes within the plant, is called mutation breeding. Okay, so in which particular category we are using mutation breeding? So wild relatives of many cultivated species of plants have certain resistance character but a very low yield. So what we are saying, that whatever the plants we are domesticating right now, they have got wild varieties. Okay, they have got wild relatives. So those wild relatives plants, right, they have got certain resistant character. They are resistant to the pest or diseases, but they have got very low productivity. Remember carefully, wild relatives of many cultivated plants, they have got the property of resistant character. Either they are resistant to diseases or to the pest, but their production is very, very low. Okay, so therefore, these resistance characters or the genes from the wild relatives can be introduced into the many cultivated plants or the species, right, which have got high yielding property. That means we are trying to combine the disease resistant character of a plant, which is from wild variety, with the high yielding character of the cultivated one. So we are trying to mix those two characters so that the progeny or the offspring will have the combination of both the parental characters. That means the progeny will carry the gene of resistant character as well as high yielding character. Clear? So let's go through. So resistance to yellow mosaic virus. Look at the point number 1. So resistance to yellow mosaic virus in okra vindi was transferred from a wild species and resulted in a new variety called Parvani Kranti. So kindly remember the name Parvani Kranti. Okay, so this Parvani Kranti is now resistant to diseases as well as it has got the higher production character. And therefore, this particular resistance to yellow mosaic virus in okra vindi was transferred from the wild species, and that leads to the production of a new species, new variety called Parvani Kranti. Then number 2, high yielding dwarf variety of wheat, Sarbati Sonara and Pusa Larma are two umber grain colored mutant produced from the red grain Sonara 64 and maximum Larma Rosso 64A respectively. Kindly remember the name of these varieties. Number 3, semi-dot variety of rice which is resistant to lozing is the product of a mutant gene by X-ray treatment. And number 4, in moong dal. Okay, in moong dal, resistance to yellow mosaic virus and powdery mildew were induced by mutation. So these are the product of mutational breeding technique. Okay, so kindly go through it. Now, what are the limitations of mutation breeding? First one, most of the induced mutations are undesirable. That we are not trying to bring those changes, but the changes are occurring, which is undesirable changes. Okay, and most of them are harmful. Lethal means harmful. Number 2, the rate of mutation is very low, and large number of plants are employed to select a certain desirable mutant. Since the rate of mutation is very low, so we have to select a large number of plants in order to select the desirable mutant. And most of the mutations are recessive. That means they are not able to express in heterozygous conditions. They will be expressed only in homozygous conditions. For this one, try to recall the cross we have done in class 10, the monohybrid cross and dihybrid cross. Clear? And mutations have tendency to revert back. And in sexually reproducing plants, mutations are inherited only if they are induced in gamete. That means in sexually reproducing organisms, for the mutations to be expressed, for the mutation to be occur, okay, and to inherit from one generation to the another generations, those mutations should be done in the gametes. That means the, in case, let's say for example, in case of human being, all right, in case of sex chromosomes XX or XY, we can say clear those changes can only be induced. But if the changes will occur in the somatic cell, in the body cell, then those mutations cannot be inherited. So in sexually reproducing organisms, for the mutations to be inherited from one generation to the another generation, those mutations should be done, should be induced in case of gametes. And therefore, the transfer of resistant genes is achieved by sexual hybridization between the target and the source plant followed by selection. So we have to go through the hybridizations and after that we have to go further selection. So these are the limitations of mutation breeding technique. Okay, now next one is plant breeding for developing resistance to insect pest. Now we are trying to develop the plant which will have the property of resistance to the insect pest, because most of the plants they are being destructed by the attack of insects, by the attack of pest. So therefore we are now trying to develop the plant which are resistant to insect pest. Okay, so insect and pest manifestations infestation is a major issue for the large-scale destruction of the cultivated crops and the cultivated plants, which will hamper the productions, which will hamper their quality. Okay, so biochemical, morphological, or physiological characteristics are used for insect resistance in host crop plants. So, what are those characters? First one, hairy leaf in several plants. You have seen most of the plants with the hairy leaves, right? So, they are associated with the resistance to insect pest. Number 2, solid stems in wheat lead to non-preference by the stem sawfly. So they are not able to attack the wheat with the solid stems. Number 3, smooth-leaved and nectareous cotton varieties do not attract the cotton bollworm, because most of the cotton they are infected by the bollworms. Okay, so smooth-leaved and nectareous cottons They are mainly do not attract the bollworms. High aspartic acids, low nitrogen and sugar content in maize provide resistance to maize against stem borer. So methods of breeding for insect pest resistance involve the same step as those for any other traits such as yield or for quality as discussed earlier. Okay, so resistant genes are made available from the cultivated varieties, germplasm collections, okay, of the crop or from the wild varieties. So some insect pest resistance crop varieties developed by hybridizations and selections are as follows. So for this one, kindly go through the page number 516, table number 9.2, where you will find the common pest resistant varieties. Okay, so first one is crop Brassica. So the Brassica is name of the crop, the variety is Pusa GUrung, and the resistance to the pest, to the insect. That means Brassica, the variety is Pusa GUrung, is resistance to the pest aphids. Similarly, flat bean. Okay, the variety is Pusa sem2 and the Pusa sem3. They are resistant to the pest, jessids, aphids, and the fruit borer. Then okra vendi. Okay, variety is Pusa sawani and Pusa a4. They are resistant to the pest, shoot, and fruit borer. Clear? So these are the name of the common pest resistance varieties. Now, next one we'll just discuss about the plant breeding for the improved food quality. Okay. Now, in spite of substantial increase in the food productions in terms of quality and quantity, more than 840 million people in the world do not have adequate food to meet their daily requirements. More than 840 million people are still they are devoid of adequate food for their daily requirements. A far greater number is not getting quality food. About 3 billion people in the world suffer from micronutrient, protein, and vitamin deficiencies. The deficiencies of nutrients in diet is called hidden hunger. Can you know this word? What is hidden hunger? The deficiency of nutrients in the diet is called hidden hunger. So these people cannot afford to buy adequate vegetables, adequate fruits, okay, legumes, fish, and meat. Diet lacking essentials micronutrients, particularly iron, vitamin A, iodine, and zinc, will increase the risks for diseases. It will reduce the lifespan of an individual organisms, and also it will disturb the mental ability of an organism. Okay, so that's why we have to introduce those programs which have been useful for the people. Clear? So today we have just learned about the plant breeding technique. So we have just learned for the plant breeding for disease resistance. We have learned the diseases caused by fungi, bacteria, and by the viruses. Then we have learned those techniques which are used to develop the disease resistant property in a plant or a crop. And among those techniques, we have called conventional breeding technique, right? Then we have learned the common disease resistance crop varieties developed in India. And for that, I have told you to go through the page number 5.15, page number 9.1. Then we have gone through mutation breeding technique. Okay, then we have given examples of few of the plant which have been developed by the mutation breeding program. Then we have learned the limitations of mutation breeding. Then number 3, we have learned the plant breeding for developing resistance to insect pest. Okay, then we have gone through the common pest resistant varieties. For that, I have told you to go through the page number 5.6, table number 9.2, and just now we have finished the plant breeding for improved food quality, where we have learned about the hidden hunger. Now, in the next class, we will do the last class for this chapter. We learn about biofortification, we learn about plant tissue culture, and we learn about single cell protein. Okay, then we will finish this chapter. Clear? So now, this was for today. See you in next class. Thank you.